we have talked about several proteins that are embedded in the membranes. For example, we have talked about receptors, we have talked about the antibodies which are initially present on the surface of the cell. So how is it that proteins get to be embedded in the membranes? We are going to look at that. That would be one of the major topics in this particular module. So let's start. First of all, we are going to talk about how the N-terminal sequence on the proteins is responsible for injection of proteins into the lumen of ER. The N-terminal ER signal sequence of soluble proteins has two functions. As we have talked about, it directs the protein to the ER membrane. It also serves as the start transfer signal or start transfer peptide that opens the pore. Even after our signal sequence has been cleaved off by specific enzymes called signal peptidases, the signal sequence remains bound to the translocator while rest of the protein is threaded continuously through the translocator as a large loop. So let's look at that. Here is our signal sequence. It was recognized by SRP, which allowed it to move to the translocator. The plug opened up, was removed from the translocator. See, notice the signal sequence is still bound to the translocator, which is keeping this structure open, and the protein is now being threaded through this translocator. After that has happened, for soluble proteins, the proteins are released into the signal peptidase. Right here is going to cleave off the signal sequence, and the protein is released into the lumen of ER. This signal sequence is also released in the membrane, which is then later on subsequently degraded by other enzymes, by the action of other enzymes. So this is what happens when we are talking about soluble proteins that are resident of ER. Now let's talk about the proteins that get embedded in the membranes. An N-terminal sequence initiates the translocation just as it does for a soluble protein, but there's an additional hydrophobic segment in the polypeptide which is further downstream from the signal sequence, which can stop the transfer process before the entire polypeptide chain is translocated. So this hydrophobic segment again is recognized, and it stops the protein transfer, and the stop transfer signal anchors the protein in the membrane after ER signal sequence. The start transfer signal has been released from the translocator and has been cleaved off. The stop transfer sequence is transferred into the lipid bilayer by lateral gating mechanism, and it remains there as a single alpha helical membrane spanning segment. Here I need to tell you about what is the lateral gating mechanism. So our Sec61 or the translocator can open two ways. It can open in two directions. Basically, it opens up like this, so protein is injected. Also, it can open up from one of its sides, which results in release of the signal sequence. And also, in our case, this protein also had a stop transfer sequence, which prevented other amino acids towards the carboxy terminus from going through the hole in the translocator. So when the translocator opens up, it not only releases the signal sequence, which has been cleaved off by signal peptidases, it also releases the protein which carried an hydrophobic alpha helix, which is now embedded in the membrane. So this is basically how we end up with proteins that are embedded in the plasma membrane. Please notice in this case, the amino terminus is in the ER lumen and carboxy terminus is in the cytosol. We can have the reverse order. We will talk about how we can reverse this order later, but I wanted to show you that this is how it is done if amino terminus is going inside the lumen of ER. Okay, so I would like to physically demonstrate what I mean by lateral opening of the Sec61. Imagine my hands are Sec61, the translocator, and this green cap is basically the plug, which is plugging this Sec61 and preventing the ions from leaking. Now we have said that once the ribosome binds to Sec61, the plug dislodges, and now the protein can go through the channel which is present in the Sec61. certain hydrophobic segments are retained by the Sec61, and once the protein synthesis is complete, these hydrophobic sequences are not allowed to pass through the translocator. They bind a place in the translocator itself. So here is the protein alpha helix, which is hydrophobic. It is going to bind a certain location in the Sec61. Remember, my hand is the Sec61, and after the protein has been completely formed, the Sec61 will open laterally, which is like this. So this protein alpha helix will now float away in the plasma membrane. So Sec61 can open two ways. One is laterally, and other opening is being plugged by that plug that we have talked about. So let's continue talking about our translocation of protein. Initially I said that generally what happens is once the proteins are being made, generally they are released with the amino terminus in the lumen of ER. So we have already seen this, how that happens. However, here I would like to emphasize that certain proteins, when they are being manufactured, they don't carry the terminal signal sequence. The signal sequence is basically somewhere inside, later down in the amino acids, which we can see right here. So when this happens, this sequence is also recognized by signal recognition particle. The same procedure happens. This protein is now taken to Sec61, and rest of the protein is then synthesized. So what determines whether the amino terminus or the carboxy terminus will enter the lumen of ER? Basically, it is determined by the charges which are adjacent to the signal sequence here. If there are more positively charged amino acids immediately following the hydrophobic core of the start transfer sequence, the signal, then they are preceding it, the start transfer sequence is inserted into the translocator in the orientation shown. Basically, the positively charged amino acids are prevented from entering the lumen of this translocator. However, the negatively charged amino acids can relatively easily get through this translocator. So here, for example, the negatively charged amino acids are closer to the amino terminus in this case, and positively charged amino acids are closer to the carboxy terminus. So this is basically how we end up with proteins which have amino terminus in the lumen of ER. Now let's look at the other scenario. If there are more positively charged amino acids immediately preceding the hydrophobic core of the start transfer sequence, then they are following it, the start transfer sequence is inserted in the orientation that you can see on the slide. Basically, again, now we have the same principle. The positively charged amino acids are closer to the amino terminus, and the negatively charged amino acids are towards the carboxy terminus, which is present somewhere right here. So when that happens, the negatively charged amino acids will go through this translocator, whereas positively charged amino acids will stay outside. So when this happens, we end up with a protein which is inserted in the ER membrane in this orientation, that the amino terminus is in the cytosol and the carboxy terminus is in the lumen of the ER. So we have seen how proteins are inserted in the membrane. We have still just talked about the single proteins which have a single transmembrane domain. Now next in the next module, we are going to see how proteins with multiple membrane passed domains are inserted in the plasma membrane.